Okay, here's a thought to start us off. Could something found in, well, what people often call magic mushrooms actually hold a key to aging better? Maybe even living longer, healthier lives.
It does sound a bit like science fiction, doesn't it?
It really does. But we're about to dig into some genuinely groundbreaking research coming out of Emory University.
That's right.
It suggests that psilocybin, the active compound in those mushrooms, might have some pretty profound effects on how we age.
And this isn't just some fringe idea.
No, exactly. This is early stage work, sure, but it just got published in a really respected journal, NPJ Aging.
Which gives it significant weight, yeah.
The findings are hinting at something, well, astonishing, really. Enhancing our physical resilience, maybe preserving the health of our cells, and potentially even impacting lifespan itself.
It makes you stop and think, for sure.
Absolutely. So, okay, let's unpack this. What's our mission for this deep dive? We're going to look closely at what this study actually found, why it's got scientists talking, and importantly, what it might eventually mean for you.
Sounds good. Where do we start?
Well, to really get into it, the Emory researchers used two main approaches. They looked at human cells in the lab first, right? And then they moved on to mice.
That's correct. They started with human cells basically in petri dishes.
And what did they treat them with? Directly with psilocybin.
Actually with psilocin, which is, what psilocybin quickly turns into inside the body. It's the active metabolite.
Got it. Psilocin. So what happened?
The results were, well, quite remarkable. They saw a significant extension in the lifespan of these cells.
How significant are we talking?
Up to 57% longer lifespan at the highest dose they used, which was 100 micromolar.
Wow, 57%. That sounds huge.
It is. And even at a lower dose, 10 micromolar, they still saw a 29% increase.
Okay, so it seems like more psilocin led to a bigger effect.
Exactly. It suggests a clear dose-dependent effect. More compound, more lifespan extension in the cells.
But was it just about living longer, or were the cells actually, healthier?
That's the crucial part. It wasn't just lifespan. They found that telomere length was preserved much better.
Telomeres, those are the little caps on the ends of our chromosomes, right? Like on shoelaces.
Precisely. They normally shorten as cells age, acting like a kind of cellular clock. Here, they stayed longer.
Okay, what else?
The cells also showed less oxidative stress. That's basically cellular damage from unstable molecules.
Sort of like rust inside the cell.
Good analogy, yeah. And they saw stronger DNA repair responses, too. The cells were better at fixing damage to their own genetic code.
So, healthier cells, living longer, better repair. Anything else major?
Yes, one more really key finding. A significant boost in a protein called ERCT1.
ERCT1, I've heard of that. It's often linked to longevity, isn't it?
Very much so. It's sometimes called a master regulator of healthy aging. Seeing it elevated is a big deal in this context.
That's a lot from just cells in a dish. A 57% lifespan boost, longer telomeres, less stress, better repair, more ERCT1.
It's definitely compelling data to start with.
But I have to ask, how much can we really take from cells in a dish? Does this kind of study always translate well?
That's a very important question. And the answer is not always. Cell studies are vital. They tell us if a compound can have an effect at a basic biological level.
But it's not the same as a whole body.
Exactly. You don't have the immune system, hormones, all the complex interactions between organs. So it's a fantastic starting point, a strong signal, but you absolutely need to see if it holds up in a living organism.
Which brings us perfectly to the second part of their research, the mice.
Right. They moved from the petri dish to live animals.
Tell us about these mice. Were they young, old?
They used 19-month-old female mice, which in human terms is roughly like someone aged 60 to 65. So definitely senior mice.
Okay, already getting on a bit. And what was the treatment? Did they give them psilocybin daily?
No, the protocol was specific. They got an initial low dose, just 5 milligrams per kilogram of body weight.
And after that?
Then they received monthly high doses, 15 milligrams per kilogram for the next 9 months.
Monthly, okay. So what happened to these older mice getting psilocybin?
Well, the survival results were pretty striking. At the end point of the study,
which was when, exactly?
They designed it to end when the untreated control group reached 50% survival, what's called median survival.
Ah, okay. So when half the untreated mice had naturally died.
Precisely. It gives a clear comparison point. At that specific time, 80% of the mice that had been treated with psilocybin were still alive.
80% versus 50%. That's quite a difference.
It really is. Now, the important caveat, as you alluded to, is they euthanized all the remaining mice at this point.
So it wasn't a study where they waited to see how long every mouse lived naturally?
Correct. It wasn't a full lifespan study in that sense, but the fact that significantly more treated mice were alive and seemingly healthier at that predefined endpoint strongly suggests they were aging more slowly up to that point.
And did they just look healthier? You mentioned seeming healthier.
Yes, there were visual observations too. The researchers noted things like better fur quality in the treated mice, darker color, more regrowth, things that often decline with age.
So physical signs backing up the survival stats.
Exactly.
Okay, here's where it gets really interesting for me. How could psilocybin be doing this? What's going on under the hood, biologically speaking?
That's the core question, isn't it?
Yeah.
The researchers pointed to two main mechanisms linking back to what they saw in the cells. First, that's RT1 protein we mentioned.
The longevity regulator?
Right. So RT1 does a lot of crucial jobs. It helps repair damaged DNA, it dials down inflammation, it boosts the cell's energy production and metabolism.
So it's like a master switch for keeping things running well.
You can think of it that way. It also promotes autophagy, that's the cell's internal cleanup crew, getting rid of junk and recycling parts.
And psilocin boosted this in the cells.
Significantly. The idea is that by increasing SART1, psilocin might be strengthening the body's own maintenance and repair systems. It's maybe like upgrading your internal maintenance crew, giving them better tools.
That's a great way to put it. Are there other ways to boost RDT1? Or is this effect from psilocin unique somehow?
That's a good point. Other things are thought to influence RT1, like calorie restriction or compounds like resveratrol. What seems exciting here is the combination of effects boosting SART1 and preserving telomeres, plus the visible mouse improvements. It suggests psilocin might be hitting multiple interconnected aging pathways.
Okay, so SART1 is mechanism one. What was the second?
Telomeres again, the aging clock.
Those chromosome caps?
Exactly. Remember how they shorten each time a cell divides? Eventually they get too short and the cell stops dividing or dies. That's a fundamental part of aging.
And psilocin helped with this.
The study found it helped preserve telomere length in the cells. This suggests the cells could potentially keep dividing and functioning healthily for longer, basically slowing down that ticking clock at the cellular level.
Okay, we need to pause here and address the elephant in the room. Psilocybin is a psychedelic drug, right?
Absolutely, yes. It's derived from specific types of mushrooms.
And it has this long history, centuries even, in traditional ceremonies. But now science is really looking at it seriously, especially for mental health.
That's true. There's huge interest in its potential for treating things like severe depression, anxiety, PTSD, even addiction.
Didn't the FDA give it some special status for depression?
Yes. Breakthrough therapy designation for treatment-resistant depression based on some very promising early results.
So it's already making waves in mental health. But this study, this throws open a whole new door, doesn't it? The idea it might affect not just the mind, but the physical aging process of the body itself.
It's a really bold possibility, connecting mental health compounds to fundamental aging biology.
It just makes you think how interconnected everything is. Who knew our cells had these tiny shoelace caps and internal maintenance crews? It almost feels like we're discovering hidden layers of how our bodies work.
And that leads us to think critically about these findings. What's fascinating here is also what the study doesn't tell us yet. It's crucial to stay grounded.
Okay, so let's talk limitations. What are the big ones?
Well, first, as we discussed, this doesn't prove psilocybin will extend human lifespan. That's a huge leap from cells in mice.
Right. And the mouse study ended early, relatively speaking.
Exactly. It wasn't a natural death study. We don't know what would have happened if they'd let them live out their full lives.
What else?
A big one is they only used female mice. We have no idea if males would respond the same way, differently, or not at all. Hormones and biology can differ significantly.
Good point. And what about using it in people?
Completely unknown territory right now. The right dose, the right timing, the long-term safety for this kind of application in humans, we just don't know.
Did the researchers themselves acknowledge this?
Oh, absolutely. They explicitly state that additional studies are warranted to identify optimized protocols for therapeutic efficacy. That's standard scientific caution.
And the classic caveat. Mouse models don't always predict human results.
Unfortunately, yes. What works in the lab or in mice often fails when tested in humans. It's a well-known challenge in drug development.
So this raises an important question. What kind of research needs to happen next to see if this potential holds up?
Well, confirming results in male mice would be key. Exploring different doses and treatment schedules, and eventually carefully designed human studies looking at safety and markers of aging.
Okay. So what does this all mean? If we try to synthesize this, what's the big picture takeaway for us right now?
I think it really opens the door to a very unconventional idea that compounds we associate with the mind, like psychedelics, might have roles to play in physical aging too.
It's blurring the lines between mental and physical health in a surprising way.
Exactly. It suggests psilocybin's potential might be much broader than we previously imagined, reaching down to the cellular level of aging.
So if, and it's still a big if, future studies pan out in humans, could psilocybin, or maybe related compounds, become part of the longevity toolkit?
Potentially, yes. You could imagine it perhaps years down the line being considered alongside other emerging approaches people are already exploring.
Like what? Things like NAD plus boosters.
Right, NAD plus boosters for cellular energy, or senolytics designed to clear out old dysfunctional zombie cells, or even highly personalized nutrition plans. Psilocybin-like compounds could fit into that future landscape.
And it's just remarkable to think that this possibility, this potential new direction for longevity science, might originate from, well, a humble mushroom growing on the forest floor.
It really highlights how much we might still have to learn from nature.
The future of healthy aging certainly seems full of surprises. This study definitely challenges some old assumptions, doesn't it? It invites us to rethink those boundaries we put up between mind and body health.
Yeah, absolutely. But again, we're not there yet. And it's crucial to stress this isn't a prescription to go out and find mushrooms for anti-aging.
Definitely not. This is early science.
But the science is indicating that something genuinely interesting is happening at the cellular level. It warrants further investigation.
So the next steps are clear. More animal studies, including males, figuring out dosing, and then very cautiously seeing if humans show similar responses.
That's the path forward.
Until then, it's definitely worth watching this space. Because sometimes it seems the path to longevity might just lead us through the forest floor. It leaves you wondering, what stands out to you from all this?